Welcome to the Rocky Mountain MS Center's webinar series, Maximizing Lifelong Brain Health in MS. Today's topic is Understanding an MS Clinical Trial Research Article, Magicians Need Not Apply. This presentation features Dr. Kavita Nair. Dr. Kavita Nair received her PhD in Pharmacy Administration from the University of Michigan in 1999. She is a professor of neurology and pharmacy at the University of Colorado Anschutz Medical Campus for the last 20 years. She is an outcomes researcher and works with the MS group in designing studies to examine differences in the safety and effectiveness of MS drugs. She has been working with the MS group since 2011 in this capacity and has developed and published several studies comparing newer and older MS drugs on clinical, safety, and patient-reported outcomes. We would also like to thank the pharmaceutical companies who have provided patient education grants to support this series. Thank you to Genentech, AbbVie, Biogen, and Genzyme. This webinar is scheduled for 1 hour. The first 45 minutes will be the presentation. If you missed something on a slide, we will be archiving this webinar along with the slides on our website at www.mscenter.org. We'll reserve approximately 15 minutes at the end to answer questions from the audience. You can submit questions at any time during the hour by typing them into the chat window on your screen. We will answer as many questions as time allows. And with that, we'll turn it over to Dr. Kavita Nair.
Thank you so much, Kelsey. Good morning. As Kelsey mentioned, I'm a faculty researcher at the University of Colorado, Department of Neurology, and my primary responsibilities are really to design research studies to help the providers that many of you see in designing studies that many of you participate in as well. So we're very fortunate here at the Rocky Mountain MS Center that so many of you as patients and as caregivers and family members are so engaged in research, both as active participants and as information seekers. So today's webinar is really to continue the discussion around research in MS. These are some of the disclosures that I want to be able to provide. So what's the goal for today? Really, I have a few objectives that I really hope to achieve by the end of this webinar. First, for those of you who are either reading a research article about a clinical trial for the first time or have been veterans at reading articles, I really would like to provide an overview of the key components that describe a research article for a clinical trial. For some of you, this can be a guide on how to interpret a clinical trial for MS therapies as you seek information on these therapies for drugs that you're either currently taking or for therapies that family members or loved ones are taking. For others, it can be a refresher. Clinical trials take several years to complete, as I'll show you in the next slide, and this extraordinary amount of information that goes into developing a drug and to the point that it has to be approved by the FDA really has to be distilled down to about 4,000 words in one clinical trial article. And so that's really a lot of information that's primarily meant for a research audience. So by walking you through today the key elements of a clinical trial, my goal is to help increase your understanding and the audience who reads these kinds of articles. Second, I really want to keep the relevancy of this webinar as close to home by using one of the new MS drugs that our providers here at the Rocky Mountain MS Center in the Department of Neurology has a lot of familiarity with, namely Ocrevus or ocrelizumab, depending on how you're most familiar knowing it. So I'll be referring to one of the key trials that was used to bring Ocrevus to the current marketplace today. I'll provide the reference for it, and so it's something that you can look up, or maybe one of the staff at the Rocky Mountain MS Center can provide some links on how you can get access to the article. You won't need to refer to it during this webinar, so we'll just refer to it as appropriate. All right. So moving on. So if you look at these terms and you go, oh, goodness gracious, am I going to be tested on all these terms by the end of this webinar? But these are the terms I really hope by the end of the webinar that you start getting to a feel of things that seem very second nature to you. So really briefly, because we're going to talk about these terms throughout the number. The first four terms, the first four bullets, if you will, these terms refer to overall the design of a clinical trial. So for the most part, when you read clinical trials, and specifically the clinical trials that we're going to talk about today in Ocrevus, you'll come across these four terms, and we'll talk about what they mean. The next two bullets, starting from eligibility criteria, informed consent, these refer to different procedures that are used in clinical trials and how they're used to really achieve the objectives. You need to have procedures in place, like things you do to cook a meal, procedures in place to achieve a clinical trial. Outcome measures really refer to what are the endpoints being examined. Why are we doing this, and what are the endpoints being examined? You have a variety of endpoints. Not all trials measure all endpoints. And finally, the last four bullet points, all the way from outcome measures to p-values, these probably refer to the areas that folks really like the least, or maybe have the least amount of information, which is how is the data being measured, how is it being presented, and what are some of the overall statistical measures and methods that are used to explain the results. And so we're going to go through some of these, or actually all of these aspects. So really briefly, before we get into the clinical slides, let me talk to you a little bit about the overview about how drugs are approved in the United States. So there are four phases really of clinical trials. Phases 1 through 3 really talk about the processes that any drug submitted needs to go through before it's approved in the United States. Phase 1, or prior to phase 1, a drug company goes through identifying several hundreds, thousands of chemical entities, or moieties as we call them, MOITIES, before they can identify one that actually shows some promise and has demonstrated safety in animal models. So the primary objective before we get to phase 1 is finding a chemical entity or a moiety that shows safety. And the phase 1 of a clinical trial is where healthy volunteers, so patients or volunteers are healthy who don't have the disease, who don't have MS, the drug is given to them to essentially ensure that the drug is safe. So phase 1 is about safety. It's a smaller phase for the most part. Phase 2 is where you start looking at patients who have the disease. In our case, we're looking at patients who have MS. And
I found something on the web. For face-to-face, where do you start with in a patient have the disease?
And there we have Siri, also confirming the fact that phase 2, it's one of our phones here, phase 2 is essentially where we look at a smaller group of patients, roughly about 300 patients who have the disease, once again to start examining the effectiveness of an MS drug, for example, and all the while ensuring that safety of the drug is maintained. Phase 3 is where we sort of get into the big examination, and clinical trials and in research articles are really the results of phase 3. This is where we get as big a sample as possible. It can go all the way from 1,000 up to 5,000. I've seen phase 3 studies that actually even look at about 10,000 patients. This is where we have patients with the disease, and they're examined for the effect of the drug on both safety and efficacy. So keep in mind that the goal of all aspects of the clinical trials is to foremost ensure that safety is paramount and then effectiveness. After this information is gathered and if the trials are successful and the patients have been safely guided through the trials, the information is submitted to the FDA, the Food and Drug Administration, a decision is made. If all goes well, the drug is approved. And then you have phase 4, which is where it becomes a lot more voluntary, which is when providers start using the drug. And as patients or caregivers, you start noticing, wait a minute, in the real world where it's not a very controlled environment, the side effects or, there's self-reporting to ensure that even in the real world where there's not a lot of monitoring, where there's not a lot of controlled situations, things can still happen. And that's what happens in phase 4. So we're going to talk a lot about the results of phase 3, and that's really the data that is used to inform decision making. So what's the value of clinical trials? Maybe this is kind of a sort of an obvious question. You're here, so obviously you think there's value to it, but I just want to briefly highlight on a few things. First, we have a tremendous growth in the number of disease-modifying agents, and I'm going to refer to all MS drugs as disease-modifying agents. Since 2010, we had about 4 or 5 agents, but I know 2010 was in my lifetime, and we have about 12 to 15 new agents, with a lot more agents expected by 2020. So as a result of that, the information is rapidly growing, and what it has done is 2 things. One is the MS disease in itself is pretty unique. You have different kinds of MS disease. It also varies by how long you've had the disease, your gender. It also differs by the kind of previous treatments you've responded to, and whether you're new to the disease or new to treatment, whether you're not new to treatment. So the growth in the number of DMTs or disease-modifying therapies that are available, plus your unique nature of your MS disease, I think really gives a unique opportunity for a patient or their caregiver to become really even better self-advocates of their own treatment. And again, I've always said this, as someone who's worked with patients across the continuum of chronic disease states, the MS patient group is an incredibly self-advocating group, and by getting information that's rapidly growing, here's another chance to be incredibly self-informed about personalizing your own treatment plan. And that's what, getting more involved in understanding the research behind these newer drugs allows you to do. Secondly, even for our providers, so for the most part, how do providers make decisions? Obviously, we really enjoy the fact that our providers are incredibly experienced, and there's no greater experience than clinical experience. But once again, to keep up with this rapid influx of new drugs, the evidence, we'll talk about how this evidence is generated, plus the clinical experience, plus your own advocacy for your treatment, allows this decision-making to be truly shared and truly tailored to your own needs. So you, and along with your neurologist, can be truly self-empowered in managing your own care. All right, so let's dive into it. There are 2 kinds of clinical studies, broadly speaking, and I just want to give a caveat. We're going to talk about clinical trials. The difference between a clinical trial and an observational study. The clinical trial is where patients are assigned, and many of you might have signed up where you're told you can join the study, you may not know what drug you're receiving. You're assigned to either get a treatment or you might get a placebo. That's a clinical trial, and those are the studies we're going to talk about today. Then you have observational studies. This is where you might be already taking a drug, and we want to see the effect of you taking this drug and some follow-up effect of that. Or you can choose to be part of a study where you already know the drug you're taking. Observational studies obviously play a large role as well. We won't talk about those. It's kind of beyond the scope of this. We're going to talk about clinical trials today. So the types of clinical trials. We have randomized clinical trial. You might see some references to it known as the RCT. So the key to remember with this is randomized clinical trial. Randomization is very important, and we'll get into it in a few slides as to why that's important. So I don't like to read off slides. We have you can see the growing list of incredibly smart people on this webinar. But in a randomized clinical trial, patients are randomly allocated into 2 groups. And for simplicity and for illustrative purposes, I'm going to assume that we are talking about a clinical trial where there's a treatment group, so a MS disease-modifying agent, and a control group. So in here, patients are randomly allocated to receive a treatment, and this is a drug of interest that we're studying, and a control group. The control group could either be another DMT, it could be an older DMT, or it could be no drug, placebo. I will say that as more and more drugs get available, the notion of comparing a new MS DMT to a placebo becomes less and less attractive, almost unethical. So you'll see fewer clinical trials that are now being designed where a new MS DMT is being compared to a placebo. In a randomized clinical trial, the 2 drugs, after patients have been randomly allocated to these 2 drugs, they are then followed forward for a period of time, and during which they're compared out into certain outcomes. So that's a randomized clinical trial. 2 groups for the most part, treatment group, control group. Control group could be a placebo or another MS DMT. They're followed forward for a certain period of time, and at the end of which they're compared to certain outcomes. For the most part, and this has been one of the criticisms of clinical trials, the follow-up period is 2 years. And for those of you on this webinar who are patients, I think you will understand very quickly and agree very quickly that MS is a disease that far exceeds the 2-year time span. We have another kind of a clinical trial. This is known as a randomized crossover clinical trial. Here, once again, patients are allocated randomly across 2 treatment groups. Here it gets a little bit different. So once patients are allocated into group A, say new treatment group, and group B, say older treatment group, after a certain amount of time, they're followed, just like we talked about in the previous slide. There's a certain washout period. This is the period at which patients are sort of washed out or from the effects of the previous drug. Then they're switched to the other treatment group that they weren't receiving and followed for the same period of time. And so the purpose of this design is to allow patients to really experience the effects of the drug that they were originally on and the drug that they were not exposed to. And it really tries to mitigate the impact of bias. I'll talk about bias in a few minutes. Okay. The article that I'm going to refer to in certain time points is the article that came out in 2017. It's the combination of 2 trials known as the opera, yes, as in the opera, singing wonderful opera singers, opera 1 and 2, that essentially compared ocrelizumab, which is a new disease-modifying therapy that was approved in March of 2017, to Rebif. So again, new disease-modifying therapy compared to an older DMT. It's an infusion that's given once every 6 months after the first dose. And it was compared to various efficacy outcomes, and we'll talk about that when we talk about different measures. So I'll refer to this article. There's a reference for it. Some of you can find it online, or we'll ask the staff of the Rocky Mountain MS Center to make it available for those of you that are interested. But I just wanted to give you this is the article I'll refer to at some points during the presentation. All right. So why randomization? Why can't randomization is complicated? It's not something that patients may or may not like. You tell me, because you don't always know the drug you're getting. It's not the easiest method to use. It can be expensive. So randomization, really, the crux of randomization is used to ensure that as a patient, you have an equal and independent chance of being assigned to either study group, either the treatment arm or the control group in a trial. And the reason we do it is to make sure that any result we get, we're trying to compare treatment group to control group, the result is really due to the drug and not any sort of external bias factors. So it's really trying to minimize the bias. So you could really say, oh, as a result of this, the patients were given drug A or drug B and was purely random. It was not because the patients choose to select themselves into a certain drug, and as a result of that selection, it affected sort of their treatment effect. Randomization is really done through several systems. Back in the day, it was done a little bit through manual systems, but now you have very sophisticated, independent electronic monitoring systems that conduct the randomization and actually even hold the code. And at some point, if there are some adverse events or a patient really needs to be unblinded, a patient is randomized, needs to be unblinded, there are ways to do that so that the safety of the patient is always maintained. Typically, clinical trials don't involve just one site. They involve many sites. So, for example, the trial that I just talked about with ocrelizumab involved close to 300 sites nationwide and about 800 patients. And the randomization doesn't just occur by the kind of drug you get. Because some of these sites are U.S. and nationwide, they could also be stratified by region so that there's good representation across smaller regions, larger regions, as well as by other factors. So if we have patients that have more severe disability, there's equal representation across the different kinds of MS disease factors by region, and so that there's a generalizability across all of these factors. Now, in addition to randomization, let me talk a little bit about the implications of bias. There are other ways to reduce bias. So I talk about bias. What does bias mean? In general, when you ask patients to sign up for a study, and we see a lot more bias in observational studies, some of these things can happen. Again, Bias is never malicious, it's always well-intentioned. You could have patients who have more aggressive disease wanting to sign up for a study because they're more aggressive disease. They want to sign up for things that could make their disease, treat their disease faster. And that could introduce an unintentional bias because you could have sicker patients in a study and the effective treatment could be influenced by the fact that you have sicker patients. And MS is a very broad disease and a very broad continuum. So that introduces bias when you don't have randomization or some of these other issues to control for randomization. The other issue is you could have older patients. And again, not to bring a stereotype, but as many of you know, when you sign up for clinical trials, there are so many visits and so many procedures and so much of your time taken, which is why, once again, we really appreciate all of you engaging in research. So you might have older patients who have lived a wonderful life and are retired or have more time to come and participate in these trials. And that may, again, skew some of the results to an older population, whereas we know MS has a tendency to affect the younger population. We also may have provider bias. I know my colleagues are very high-spirited and we all have our personal preferences and the kinds of drugs we use. And so if they know their patients are in a certain trial, may actively or with the best intention talk about certain drugs, treatment drugs over other drugs, and that can influence how a patient feels about a drug. So all these are examples, for example, of unintentional bias that can change the way a treatment is influencing or the outcome. And so in trying to minimize that, you really want to know the treatment, the effect you're seeing is really because of the treatment and not some of these factors. There are different ways to do that. And we'll talk about some of these ways. So one of the factors is blinding or masking. And what it really does, it's a little bit like the Batman approach, where in clinical trials, blinding or masking, all members of the research team are blinded. And so that means the investigator, you probably have seen that there's always a neurologist or a provider who's in charge of the research. The investigator, the study personnel, everyone is sort of blinded from knowing what drug the patient is taking. And that's not to say that everyone is literally walking around with blinders on, but it's to prevent these kind of conversations from happening so that the study is conducted and it's really just a few people like the dispensing or the research pharmacist who knows what drug you're getting so that there's no conversations about bias. There are different kinds of blinding. In a single blind, single mass study, the patient is not told what they're giving, but the research team could know. And again, there are different reasons as to why a single blind is done, particularly if there are more safety concerns on the drug. In a double-blind study, neither the patient knows nor the research team is told what the patients are giving. Only the dispensing pharmacist, because the drug has to be dispensed, knows what the patient is getting. So no one knows. This formulation of a study has probably the least amount of bias, is the most controlled. Higher burden, more expensive to do. Double dummy. Who knew research could have double dummy in it? Is used with two different treatments and different routes of administration. The study that we're talking about, and we'll get into that in a minute, is used when the treatment group and the control group have different ways in which they're consumed. One is an infusion, one is an injectable. So really blinding is a little hard because if you're getting an infusion, you know the kind of drug you're on. So how do you mask that when the routes of administration are different? However, patient safety and patient protections are always the primary concern of everyone involved in a research trial, which is why if there's ever a reason to unblind a particular patient because of safety concerns, there are ways to do that, and that's always a primary concern. Nothing is more important than patient safety. So bear with me. This is an actual study trial for the ocrelizumab versus Rebif study, and it has everything in it. This is like, hey, I'm teaching you how to cook, and now we're going to make a gourmet meal out here. Nothing like jumping into it. So I'm going to be really cautious in how I talk about this because there's a lot of information to gather. And again, I wanted to talk about things that products that you, or MS-DMT therapies that you're actually taking. So let's see if we can go through some of these terms because you just heard it about 10 minutes ago. This is a multi-center randomized, we talked about randomized, double blind, boy, we just heard that a minute ago, double dummy, just heard that a double ago, and no one's a dummy here, parallel group study. So what does this mean? And again, I'm not going to quiz you on this. I'm just going to go through this to give you a sense of what this study design looks like. And I want to apologize in advance that it is one of the more complicated designs, but it also represents a study design for one of the products that many of you are either familiar with or taking or know loved ones that are taking this drug. So this clinical trial involved 800 patients in total. 2 drugs. The treatment group is ocrelizumab or Ocrevus. It's an infusion given every 6 months. The first dose is a split dose given on day 1 and day 15. And the comparison group is Rebif, which is given as a self-injectable. Patients were randomized. Remember we talked about it, randomized in a 1:1 ratio, which is for every 1 patient of ocrelizumab, there was 1 patient of Rebif. Now, I say 1:1 ratio because sometimes you can do a 2:1 ratio, different ways to do it, but this part they kept thankfully simple, 1:1 ratio. Again, this was not a placebo. The control group was not a placebo. The control group was an older disease-modifying agent. Rebif has been around for 15 years. Ocrelizumab has been around for, gosh, I think we're getting into almost 18 months. So just to give a comparison. Again, the use of placebo is now ethically indefensible due to established therapies. This was a multi-center worldwide, almost 300 sites worldwide. And they did 2 trials. This is a little interesting. They did 2 trials. They did Opera 1 and 2. Patients were recruited between 2011 and 2013. The trials were non-overlapping, but they were done separately, and the data was presented as a combined data. So maybe the folks at the sponsor was Genentech, just like the Opera. There was no real rationale for doing it separately. All right, so we talked about the multi-center, 300 sites. We talked about why randomization, right? Let's talk about we talked about the double blind. That means at every site, almost 300 sites, all the investigators, there was a neurologist at every site who did all the procedures. We'll talk about the procedures. And everyone, with the exception of the dispensing pharmacist, because patients had to actually get the drug, so the pharmacist dispensed, really didn't know what the patients were receiving. And you go, well, how's that possible? One drug is an infusion, and one drug is a self-injectable. I think we know the difference. And I go, well, I'm no double dummy, but we'll get to that right away. So the patients were given ocrelizumab as a dual infusion of 300 milligrams on day 1 and 15. For the first 24 treatment cycle, the study was actually conducted for over 26 weeks. And I'm going to use my nifty little, so here, till here, they were given ocrelizumab as an infusion, or they were given Rebif out here, as you can see, right? And they were given either infusion or they were given Rebif. And this was an infusion and this was a self-injectable. In addition, they were also given in parallel, so they received either of these drugs as an active group. They were also randomized at the same time to receive dummy infusions. So this group received a dummy infusion of Rebif, which is a dummy infusion. It was a self-injectable. They were just really saline. And this group received a dummy infusion of ocrelizumab. Let me take my nifty little, here we go, dummy infusion here. Oops, yeah. So these groups were really receiving both drugs out here, but they were receiving the active group with the active ingredient as well as a dummy version of the comparator group. So now you go, oh, I see, that's where the double dummy aspect comes into place. And why would they do that? So now patients don't know what they're getting because they're getting both. They're getting an infusion. They get infusion as well as they're getting a self-injectable. Only one of these has the actual active drug in it. And they got this all the way for 96 weeks, which is roughly 2 years. And the dummy version of what they were getting looked identical to what a actual ocrelizumab infusion would be or an actual Rebif injectable would be. So you can see that in this very, sort of spy, James Bond kind of a design, there's a higher burden on patients, there's a higher burden on the study design itself a little bit, there's a higher burden on the investigators, but it really tries to eliminate as much of the bias as possible. And then when you look at the study design, and the reason there's a parallel groups term in here is because both groups of patients were in effect receiving both the active drug as well as a group, and it was in parallel. So that is a study design of a drug that we use quite often here at the Rocky Mountain Center, and how it falls into and why it's done. It's not, I don't think it's necessarily because all these terms in one sentence or one title make it seem fancy. Okay, now I'll move on. Right, now let's talk about the procedures. So they've designed this study. These are probably the aspects that you folks are the most familiar with as well. We have inclusion criteria. Why do we have inclusion criteria? Well, not everyone can join a trial, although we really appreciate everyone who signs up for the trial. The reason is, again, primarily safety. With the treatments that are available today, the characteristics of the patient have to match what the treatment is designed for. So not every patient who has certain forms of MS, some of the patients who've had MS for a long time, aren't necessarily tailored for some of these treatments. So for example, MS disease history matters. If you've had a relapse in the last 30 days or the last 6 months before starting a trial, that can affect or alter your ability to participate in a trial and even make it ineffective for you. Your level of disability. So the more disabled you are, you may be better qualified for other trials. So the inclusion criteria is really a measure to ensure two things. One, that patients who are fairly similar in their MS disease presentation are included. And that way, when we get the results of the trial, we can generalize and say, generally the patients between this age and this age range who have this level of disability, who have this kind of a previous MS history, the drug works, the treatment is best served for this population. Again, as patients and as caregivers of patients, you're well aware that the MS course history can be pretty diverse. Also for safety, if there are MS disproportionately affects women, so we certainly want to make sure that women who are trying to get pregnant are safe, because many of these drugs have yet to have safety proven during pregnancy or on the chance you get pregnant. So inclusion criteria, like exclusion criteria, really ensures that patients are similar so that the effects of the treatment can be generalized to the MS disease population that it benefits the most. It's safe for patients, and it's also safe for patients who not only have a similar MS disease history, but also doesn't affect patients who have had other kinds of conditions. So, for example, many of the newer MS drugs really affect the immune system and depress the immune system, and so if you've had other conditions that can have affected your immune system, like prior cancer treatment or other drugs, for example, to treat rheumatoid arthritis and things like that, taking some of the new MS drugs or being part of a trial that includes some of these MS drugs could worsen or could make that issue even worse. So this inclusion and exclusion criteria are designed to really ensure generalizability and safety. These are procedures. We get into informed consent. Boy, this is the part that I think patients are really, really designed to make it safe for patients. Most important thing to know about informed consent, I think you folks know this, is that participation in a trial is always voluntary. It is a privilege for us when patients agree to participate in a trial, and it is your privilege and your choice always to leave a trial. And it is our responsibility to always inform you fully and thoroughly, as many times as we need to, on the purpose of the study, what the study entails, what the benefits and risks are, so that you as a patient are fully informed as to why what the study entails, why you should participate, what benefits it could be for you, and what risks it are. These are not easy procedures, and again, we are very grateful at the Rocky Mountain MS Center that our patients are as engaged as they are. And for those of you who are tuning in and are not necessarily part of the Rocky Mountain MS Network, I cannot tell you, your participation is why we can do the research and continue to advance the science of MS. So these are procedures, and for us, for informed consent is not necessarily just a sheet of paper with things we write and make up. The informed consent process has to be approved by an institutional review board. The language is very specific, so it's something that goes through extensive review before we can talk to a patient about it. Trial procedures. So this is probably my least favorite slide because it's so busy, but this is one of 3 pages or 3 tables of the trial procedures that I took from the ocrelizumab trial. And this is where really this is the burden on the patient. Every trial has different processes. You can see out here, I'm going to take my nifty little, we do the informed consent, and these are all the different kind of procedures that we do. Procedures are essentially the tests, the assessments that we do to collect the data. And they can vary. You can see out here that they vary. The trial procedures really vary. They go all the way to 96 weeks, and you can see out here how they vary and how often you come and do it. So this is when patients come into the clinic or come to different sites. They may have to go to different sites to get an MRI, to engage and get different tests taken, different physical examination, get different labs drawn. This is the data collection. This entire trial procedures is the heart of the trial. And if you do get a chance to go look at the trial that I was referencing, there are 3 pages of it. How this is done, who does it, how carefully it's assessed, how much time you spent. A big part of whether you want to participate in a trial or not is understanding how much of your time this takes to do this, and how this can be. We try to make this simpler by having you, some of these tests, depending on the complexity of the trial, can be maybe rolled into some of your regular visits with your neurologist. But again, in this trial, this is one of 3 pages of tests and procedures that you would participate in if you did this trial. Okay, length of the trial. Generally, a trial takes about 2 years. You have a screening period where you come in to see if you're eligible. Even if you meet age criteria and MS disease criteria, you may or may not have other conditions, other prior conditions, prior immune responses that may render you eligible or ineligible. There's the entire treatment period. Generally, trials take about 2 years. There's a screening period before, and then there's a follow-up period. So after you finish the 2-year period, you may have a safety follow-up period as well. So trial commitment can take easily about 2 to 3 years, and a lot of studies or a lot of trials will have a follow-up period for 48 weeks or an open label period where they like to follow patients, because the more data we have on patients, the more we know about the long-term safety. You know, these are very powerful drugs that we are introducing in clinical trials, and the longer the data we have on it, the more we know about safety. So something else to think about that, more is more in the case of the amount of time spent in the clinical trial. All right, I'm going to move along a little bit quicker because we have only a few minutes left. Let's talk about endpoints. We have relapse rates. You folks are familiar with relapses. Unfortunately, many of you have to suffer through them. But in trial endpoints, relapse rates aren't necessarily about relapse. Think about relapses the way you guys conceptualize them. But for the most part, a relapse rate as a trial endpoint is when you have worsening symptoms that last for about 24 hours, but also are co-validated by an increase in the EDSS. Now, I'm not sure how many of you are familiar with the EDSS from a standard of care clinic standpoint, but this is what an EDSS is. It's a physical disability scale. And for every trial, almost every MS trial, it's a standard measure of physical disability. Some of you may or may not know what your EDSS scale is. So if your EDSS scale is here, your fairly minimal disability. As your EDSS disability scale increases, the number increases, your disability level increases as well. So when your relapse rate, the number on your disability scale increases by either 1.0 or 0.5, as measured by clinicians, so you have your clinician measure it through it's about a 20-minute procedure, then you're defined as having a relapse rate or a confirmed disability progression. So this is a scale that is used primarily in research trials to determine is a patient progressing in terms of their disability progression. One of the disadvantages of this is, as you folks know, MS is far more than physical disability. There's lots of other issues with disability progression, but that's for a different conversation. Okay. Other endpoints, we talked about confirmed disability progression. Right. I'm going to wrap up here with some important aspects about data analysis. I think this is the part that's least understood. First, we want to look at baseline data. Even though we try to get patients that look very similar in trials, at the end of the study, we want to know who stayed in a trial. We talked about how long the trials are. So when the trial is completed, 2.5, 3 years later, we look at the two treatment groups. How comparable are these groups? Did all the people who dropped out or all the people who withdrew from the trial, was there something systematically different from them? Are we at the end of the trial still getting comparable groups? Then we look at two different ways to analyze the data. Let's assume we started with, as in the ocrelizumab trial, 400 patients per group. Did we end up with 400 patients per group? And if we didn't, do we analyze the outcome measure looking at all the patients who we started off with? That's called the intent to treat analysis. You'll see that term. So the analysis of primary outcome includes all patients randomized to each treatment group, whether or not they completed study protocol. Patients could have dropped out. Patients could have been forced to withdraw because of safety reasons. Or do we do a completer analysis, which is, we started off with 400 patients. By the end of 3 years, we lost 50 patients. Do we only include those patients who were randomized to each treatment protocol but who completed the study protocol? So different trials will do it differently, all of which are very valid ways of doing it. All right, we're in the home stretch here. Bear with me a little bit more. Okay, p-values. We hear this all the time. So what's a p-value? Everyone talks about statistical significance. So first I want to talk about effect size. So let's assume we're looking at annualized relapse rates, right? That's one of the outcomes. What are the relapse rates different between the treatment group and the control group? The difference is known as between the treatment group is known as an effect size. An effect size is that size of a difference which we deem as clinically meaningful. It's just a statistical term. The effect size really is related to the outcome. So an effect size for a relapse rate is going to be different than an effect size for a difference in blood pressure. It varies. The term itself is statistical. The clinical meaningfulness of it varies depending on the discipline and what clinicians consider meaningful. The p-value indicates that any difference between two groups can just occur by chance. Two groups are different. You and I are different. And the level of significance defines whether the probability is because of chance or the probability is because the difference is truly statistically different because, wow, these groups are really different. That's really what a p-value is. So p-value indicates the probability that the difference between the study groups simply occurred by chance. In fact, if there's chance, then there's really no difference. We just happen to be different. The level of the p-value means that the probability is less. If it's a p-value of less than 0.01 and a p-value of less than 0.05 means that the probability is statistically significant. That's all there is to it. People get very hung up on p-values, and there's a lot of debate right now as to who chose this. Why could it be something a little different? So that's what a p-value is. It's simply a number, and we've been using this as a norm. Okay, we're ending here in a couple more slides, and we'll get into questions. This is the results from the Hauser trial. I think I told you they combined OPERA-1 and OPERA-2. We're now getting into something, the annualized relapse rate. So this shows us the annualized relapse rate at the end of 2 years was 0.16 for ocrelizumab and 0.29 for Rebif. So this tells me that this is lower than this. But what are confidence intervals, right? So confidence intervals is the range within which the effect of a treatment is likely to fall. The narrower the interval, the greater the accuracy of the estimate. So if we go back here Here's a confidence interval for ocrelizumab, and here's a confidence interval for interferon. This range is narrower than this range. So this tells me that the accuracy of this estimate for annualized relapse rate is likely to be more accurate than this estimate. And if you look at the p-value, the p-value is far greater than 0.05. So I think this difference, this number compared to this number, shows that it is statistically significant. That's what I wanted to point out about confidence intervals. Confidence intervals and p-values are two different ways to determine the accuracy of this estimate when compared to the accuracy of this estimate, and whether these numbers here, whether the difference has any statistical significance, as we've traditionally defined it. I'll end with this so that we don't take more eat into your question time. Rate ratio is the rate of an outcome in one treatment group divided by the rate in another group. Going back to this slide, another way to talk about the annualized relapse rate is the rate ratio, which means the rate of an annualized relapse rate in the ocrelizumab group is almost half of that as in the rebif group. It's the outcome of this group divided by this group, which kind of is reflected here. If you look at it here, it's also kind of half, it's about 0.16, 0.2, and it is statistically significant. So I wanted to get to that. So at the end of the day, I hope this helped. And with that, I will, here's a number if you want to get involved in any research. I will take any questions.
Wonderful. Thank you, Kavita. So we do have a couple of questions that were submitted ahead of time, and we'll start off with those. Anyone in the audience, you're welcome to submit your questions using the chat window, and we will move to those next. So we have one question talking about how to discern risk when you're looking at clinical trials based on other things that might be going on with their health, or how do we translate trials into what the real risk is going to be of them?
Okay, excellent question. So one of the things I didn't touch upon really is we talked about endpoints, really talked about some of the efficacy endpoints, but another huge category of endpoints is the safety endpoints. We do look at safety endpoints. We do look at the rate of adverse events. We do look at the rate of serious adverse events. So I think the question was, how do you translate risks in clinical trials? So my suggestion would be to, just as we have given methods to understand the probability of how you look at efficacy endpoints, the safety endpoints are given similar statistics, maybe not as detailed, because safety rarely tends to be a primary endpoint, but to look at the numbers and to keep it in context. And then most importantly, talk to your provider about it. Because safety is important, but to talk to your provider about it and to look at the data with the same lens that you would look at the efficacy data and say, this was the annualized relapse rate. One is less than the other. How can I use that perspective to look at the safety data? And then talk to my provider about it and say, okay, there are 3 rates of this infection. Should I be worried? But there were 800 patients. Does that help?
Yeah. And then also, are the pharmaceutical companies funding these trials, and what biases must one take into account there?
Okay. Excellent question. So the first part of the question is yes. The pharmaceutical companies are funding these trials. In fact, I remember this estimate that came out several years ago that on average it costs $800 million to fund a trial. So it's a mixed response, right? We also have PCORI, which is the Patient-Centered Outcomes Research Institute, so PCORI, and it's a national agency that was developed about 10 years ago from grants, funding from the ACA. So we also have independent agencies that are funding these trials as well. In fact, our own Dr. Corboy has a trial funded under PCORI. So if for those of you looking for a trial, please contact the Rocky Mountain MS Center to enroll in the trial. So the biases are that, yes, we have companies that are funding it, and obviously they have products that they're funding the trials for. I think the upside is the reason we get into these very complicated randomized design is exactly to try to avoid some of these biases. because when you're also the investigators participating in these studies are investigators like the Rocky Mountain MS Center, University of Colorado, and so as investigators, our duty is first and foremost to the patients and to good research. But money doesn't grow on trees, and we want newer treatments. So hopefully that balances it all out.
We had a more general question come in over the chat system. What is the meaning of the term disease-modifying therapy, and how is it different from any other treatment that uses medication?
Excellent. So disease-modifying treatment is sort of the general term that's used to refer to MS agents. And so we say DMTs, that's kind of our little quick abbreviation. So it's a general term that's used to refer to any MS agent, and we use it very specifically for MS agents. So anywhere from the older MS agents to any newer agents, we broadly use the term disease-modifying agents or DMTs, disease-modifying treatments. I hope that clarifies the response.
So how big should a study be for someone to consider the results valid?
Excellent question, and I wish I touched upon it. So one of the key aspects of designing a trial is determining statistical power. And power refers to what is the sample size or the size of the study that is needed to truly determine the statistical effect size. Remember we talked about effect size, the difference between the treatment group and the control group to generate a meaningful difference. One is clinical meaningful difference. It's different for hypertension or cardiovascular disease versus MS disease. But the more important issue in clinical trial is a statistical difference. In order to see a 10 or 20 or 30% difference in response rate for the annualized relapse rate, we have to know how to get whether we need 800 patients, as in the trial we talked about, or we need 2,000 patients. And there are power calculations that look at previous data, that look at different combinations of statistical and clinical factors. And those are key to determining what the sample size is and therefore how big a trial is. Without that, a trial cannot be designed appropriately. So that's the data that's used to determine how big a study is and therefore how valid it is.
And then what are some other key things that you want to look for when trying to tell if a study is legitimate?
Okay. I think trials, for the most part, so the things we look at, right? I think it would be, we really want a trial to be compared to other DMTs, so, comparison between two DMTs. I think we want to make sure that as the inclusion criteria were as representative as possible, so MS diseases that are, people that are included that are, over the range of people have MS, we have patients that are over the age of 55. It'll be nice to see, these are things, it's not necessarily legitimacy, but we'd like to see more progression in this area. I think we would like to see the fact that, we have lots of newer agents. We'd like to see more comparison between some of the newer agents because people are progressing more towards using more newer agents. And I think for the most part, in terms of legitimacy, is we'd like to be able to know that the studies were done, and even though we have to have inclusion criteria to make sure that the patients are as similar as possible, so that you can generate results, it would be nice to see a little bit of subgroup analysis. So what about the women who maybe got pregnant during the trial, or the group of people whose disease progressed? The trials are complicated. They're across several countries. They're across several sites. You can't account for everything, but there's always a little subgroup analysis so that particular subgroup may apply to me as a patient. And it'll be great to see some of that subgroup analysis to know that even though this is a broad study, this particular population, what about patients whose disease got aggressive after 40? That could be me. Every little bit of information counts. And I'd love to see more information about pediatric populations as well.
Absolutely. We did have one question come in through the chat window about specifically some new research. And I just wanted to let you guys know we have several presentations on our website, mscenter.org, that go over research updates. Particularly in May, we did a presentation at our Education Summit that you can find on our website. We will be doing another education summit on November 3rd, and we will have another research updates presentation, as well as look in December, we will be doing a 1-hour webinar discussing research updates. So we're not going to go into specific research today, but you can find lots of that on our website. And I think that we might be running out of questions as well as time, actually. If anyone has any last-minute questions to submit. Okay, thank you, Dr. Kavita Nair, for sharing your time and your expertise with us. That hotline is on the screen now if you're interested in becoming involved in research. This website will be archived on our website, www.mscenter.org.
Thank you.